Welcome to the Jack Westin MCAT podcast with your hosts, Michael Maleka
and Molly Kilty. Hello, everyone, and welcome back to another episode of our Jack Westin MCAT podcast with your hosts, Mike and Molly. Mike, our last episode was a pretty big one. I think it was pretty anticipated, if not by our listeners, by us.
Yes.
Because we got to go over the basic findings of AMC Exam 6. That was a lot of fun, especially with all the data that we got to recruit. Thank you again, content team. You're the best.
They're great. Yeah, no, it was super cool, both taking it and then also like reflecting on it, both from a personal experience, but also on the data to kind of see like, okay, how did the personal experience actually relate to the data? How did like, oh, I felt it was this way, but oh, actually the data is showing this. There were a lot of similarities there. And it also, again, was really, really great to get some insight as like the direction the AMC and the MCAT's going. And especially going into 2026. And so I think if anyone's listening to this and didn't have a chance to check out that episode, highly recommend checking it out, especially if you intend on taking AMC 6 at all in the future, especially with tests coming up. But this episode, we're kind of like getting back more to like kind of what people expect from us, right?
Our regularly scheduled programming.
Exactly, right? Where we're going to be kind of diving into, I think, as Molly and I are both avid gym goers, we love the gym. One of our kind of favorite topics, which is muscle contraction. We're going to be talking about how movement actually happens. This is something that's really, really important for the MCAT. I think it's going to be really, really cool. I know Molly has some great analogies that I make fun of her of, but they're actually really, really good about, how does movement actually happen? A lot of times we find that students kind of learn these parts, there's going to be buzzwords like the cell, like about the, why am I forgetting words now? About the cycle.
Neuromuscular junction.
Neuromuscular junction.
Cycling.
This crossbreed cycling. I was like, why am I forgetting the word crossbreed cycling? But crossbreed cycling, my goodness, I'm spacey today. But crossbreed cycling, neuromuscular junctions, calcium release, the sarcoplasmic reticulum. We learn all of these things in isolation. But what we're going to do is to actually take you through the process from actual initiation by the neuron to muscle contraction, what actually happens. Because the MCAT, it kind of reminds me of a little bit about metabolism. The MCAT loves throwing like wrenches in that pathway. They love saying, hey, we got rid of this part, we changed this part. What is the downstream effect? Like they're not testing like these little details. So just like what we did with metabolism when we broke down metabolism, the steps, the order, we're going to be taking it that way today and actually breaking down the steps involved in muscle contraction. So I'm really looking forward to talking about this.
Yes. And it really is it's a story, right? Mm-hmm. It's like all the way from your motor cortex and your frontal lobe, all the way down to the actual contraction of your muscles, a skeletal muscle in that case. Mm-hmm. It's all one cohesive story, right? There's multiple chapters, but it's all one thing leads to the next. It's a domino effect. So I'm excited to get into it. And we're going to kind of start with the basics and dive in deep pretty quickly. That being said, most of what we're going to talk about today is about moving your actual skeleton, right? Like that's what I think a lot of people are very interested in. It's obviously very visual. You could see it happening, but there's actually multiple types of muscle, right? And there's actually a lot of ways that you could classify muscle cells and by their function, but let's start with the basics. Okay. We're mostly going to be talking about the type of muscle that moves our skeleton, which is given a very logical name, which is skeletal muscle. I know, shocking. And I think a lot of the properties of skeletal muscle make a lot of sense, right? Like, of course, you are, for the most part, in control of the movement of your skeleton, right? This is a voluntary muscle fiber, right? And you also know that in order to move your skeleton, which is very dense and heavy, right? It makes up a huge chunk of our body weight. You also have to have a lot of force in the contraction, especially if you are like Mike and I, and you love to go to the gym and lift weights and run. Just kidding. We hate running or whatever. Your skeletal muscles have to be very powerful in that contractile ability. Okay. So when we talk about how to tell if something is capable of very forceful contraction, one of the properties we look at is called striations. And we're going to talk about what striations mean on a more micro level. But for now, if your muscle fiber, looking at it, you almost see stripes
Hmm.
Basically the alignment, the nice even alignment of all of the contractile units. It just creates more force. Okay. So skeletal muscle, we're voluntary and we're striated. And I think both of those make sense
Hmm.
because we need to be able to generate pretty extreme contractions. Okay.
Hmm.
Some of these properties are shared with cardiac muscle. We also need to generate quite a bit of force in our heart to pump blood all over the place. We talked about fluids, I don't even know how many weeks
Long
ago.
time ago, months ago at this point.
Yeah.
Uh,
But, when we talk about fluids, you recognize that in the context of the cardiovascular system, your heart is what's creating the force to move all of your blood, especially if you think of blood going down to your feet. The force of your contraction needs to provide enough pressure to get all that blood back up through your veins back to your heart, right? Like it has to be able to contract very strong. So cardiac muscle is also striated, just like
mhm.
skeletal, although you don't want to have to voluntarily control that one.
That would be awful, right? To have to think like, okay, pump,
Beat,
pump,
beat,
but
beat,
like each and
beat.
every time. So there's this kind of cool thing that the heart has. It's actually really, really unique, which are these kind of like leak channels, right? These calcium leak
Mhm.
channels that allow that like automatic beating. Because normally with skeletal muscles, yeah, I don't want my muscle to just happen on its own. My arm would be failing, right? But we need the our heart to be able to be a little bit more self-sufficient. So that's when we have these calcium leak channels that allow it to get up to its potential, beat, get back down, but get up on its own without it. So again, a lot of similarities, like you said, Molly, like a lot of similarities to skeletal muscle, but a few differences. They are kind of striated. They have what are called intercalated discs, but they're not so lined up like a skeletal muscle is. They're more like jagged, kind of all over the place, not as coordinated. But right, they have these kind of calcium leak channels that allow for that automatic beating. But speaking about automatic, that kind of brings us to like the last form of muscle, which is our smooth muscle. Now, this is the kind of muscle where it kind of has the same function as the cardiac muscle in that we don't want to consciously think about it. Because imagine how many things in your body that are happening that involve muscles that you don't have to consciously think about. Think changing your blood vessel diameter. Think, for the most part, breathing. You don't have to think about it usually. Think about, anything, again, your gut, in your digestive system. There are muscles there as well. Whenever there is muscle where you don't actually have to think about it, right, that is involved in smooth muscle. So it's involuntary, and as well, smooth muscle doesn't really have to contract as hard as skeletal muscle. The skeletal muscle, we're lifting really, really big weights. So in that way, smooth muscle doesn't have to have that really forceful contraction, so they don't have any striations. They don't have this like lining up of sarcomeres, so they are non-striated, we say.
Yeah.
So striations, skeletal muscle. If you have these intercalated discs, kind of jagged cardiac muscle, no striations at all, smooth muscle. That's kind of like just a quick way to visually distinguish which muscle you're looking for. Right. And
Yeah.
so, like we said, like these ones are much like smoother contractions, much slower contractions, not as forceful as the skeletal muscle. And I think that's kind of like the kind of main three, at least the MCAT likes to classify our different types of muscle on this exam.
Yeah. And I think it makes sense when you think about smooth muscle, you don't want it to be striated. Like just imagine smooth muscle, yeah, lines your blood vessels. Think of powerful contraction, what that would
Squeezing your
do.
blood back.
Squeezing your blood, like ouch. And also think your entire GI tract for the most part
Mhm.
is lined by smooth muscle.
Mhm.
Imagine you're trying to, let's say, swallow something,
Mhm.
right? Peristalsis in your esophagus or
And
in
then
your
just
stomach
explode.
or in your, and it's just
Right.
crushing like
Choking every single
literally.
meal.
Oh my gosh, that would be horrible. Yes,
Mhm.
smooth muscle, also non-striated.
Mhm.
And yeah, this is basically the overview of muscles. You could dive in deeper, especially with skeletal muscles.
Mhm.
Cardiac, of course, you can get into heart function. You could talk about, the sinoatrial node and the atrioventricular node and how that coordinates heart contraction and the electrical, system with the Purkinje fibers. But that's more heart anatomy than
Mhm.
just about the muscle cell itself. With skeletal muscle, you could also get into like more exercise science with like type 1 fibers and type 2A fibers and 2B. But really, for the most part, you got to know your three muscle types. And
Mhm.
from there, it's really zoomed in on a more micro level. What is going on? What is creating the contraction? That tends to be the primary area with MCAT scope that we're going to talk about. So let's get into this story. Okay, let's talk about it because there are There are a couple distinct chapters. When we talk about what happens in the motor neuron, when we talk about what happens at the neuromuscular junction, and then even when we talk about the actual muscle, we talk about how there's almost like an action potential in the muscle,
Hmm.
which then facilitates the interaction between actin and myosin, right? There's steps that have to happen before they can interact. And then once they can interact, you talk about how it actually shortens. So there are kind of these distinct chapters of our story. Let's start off at the beginning. Let's talk motor neurons. We
Love
talked
it.
about we talked about the nervous system a very long time ago. Let's do a little recap.
Right. Right. So the motor neurons. So to begin, when we talk about, contraction, we know every contraction has to begin at the brain because you actually have to tell the muscle, you have to actually intend for the muscle, the skeletal muscle, to contract. And so a signal travels from the brain to the muscle via what are called motor neurons. Again, these are cells that are always traveling away from the central nervous system, the brain and the spinal cord, delivering information to some target organ. In this case, we're talking about motor neurons targeting muscles, but motor neurons can target anything. They can target organs, they can target, anything that's just away from the CNS. We call it motor neuron. In this case, we have the motor neuron. It starts and it travels via the axon. It has a body, a cell body at the head, and it uses its long axon tail to deliver an action potential. And it's this long axon that can be many meters long, very, very long, to whatever target muscle that we're talking about here today. Let's talk about the bicep, right? We love the bicep. We know that one's pretty close to the brain. So a signal comes from my brain and travels via a neuron, right, down the spinal cord, synapses onto some other neuron, and then travels to my muscle. But now we have to get that action potential from the neuron to the actual muscle cell. Kind of a note about how it's traveling, about the action potential itself. It's using a lot of sodium and potassium channels. I don't know how deep we want to go into the action potential today, because again, if you really are like, oh my goodness, like action potential stuff is like really, really scary, neurons really scary, I really recommend like going to check out a nervous system episode where we really, really break it down. But kind of overarching, it's using these sodium and potassium channels to deliver that action potential to that target muscle. But then something happens at the end because we need to get that signal from the neuron to the actual muscle. Now, this is where at the axon terminal, like the end of that long axon, we have the axon bouton or the terminal, where instead of sodium and potassium channels that are the real important one, at the end we have these voltage gated calcium channels. Now, we call them voltage gated because they activate via a change in membrane potential, which is when action potential is. And once the action potential gets to the end, right, you have this intake, you have these calcium channels open up, and then a bunch of calcium enters the neuron. A lot of times I notice when a lot of people talk about calcium in muscle contraction, they're so focused on inside the muscle cell. But no, calcium is not only involved inside the muscle cell, it's also involved in the neuron that activated that cell. Calcium comes in, and calcium, super crazy, the signaling molecule. It's so, like when you got calcium, things happen when calcium's there. So once that calcium comes in, it causes these little packets of neurotransmitters. There's these little vesicles of neurotransmitters in the axon terminal to say, hey, calcium's here. I got to release these neurotransmitters into the space at the end after the axon terminal. So calcium comes in, and all these little packages of neurotransmitters are released out of the axon terminal into what we call the neuromuscular junction, which is that, think of it like a synapse for neurons and muscles. It's kind of like, because synapses are normally like neuron to neuron. Now we've got neuron to muscle. It's the neuromuscular junction. Makes sense. So now we're at this point where we've just released the contents, these neurotransmitters, and because we're talking about muscles, this neurotransmitter specifically is acetylcholine, right? That's like the neurotransmitter of muscle contraction. So now we've got all this acetylcholine in the neuromuscular junction. Great. Well, Molly, now what? Like, well, we got all this acetylcholine here. Great. I'm there. That's kind of part one from like, okay, we've released them
Yeah.
all, but now what do we do?
Yeah, that's chapter one of our story,
Yeah.
right? Let's go to chapter two. Okay, so we're very focused on the motor neuron and what's going on. Like you said, we walk through the whole detail of how action potentials start and ultimately end in our nervous system episode. But Motor neuron, it's done. Okay. Its whole function is release acetylcholine. Now it's the muscle chapter, right? Like
Hmm.
how does the muscle respond to this? And like you said, it's all about acetylcholine, which is that neurotransmitter that travels through the neuromuscular junction. Now, when we talk about neurotransmitters, each one actually can have many functions.
Hmm.
Acetylcholine, though, when you hear that, I want you to be thinking that's the one that's associated with muscle contraction. It's one of the hallmarks, one of the main things we want to know about this guy. So it's going to be released from those vesicles in the motor neuron, right? From that little end plate. And it's going to travel across that little cleft and it's going to find acetylcholine receptors on the muscle fiber itself. Okay, so it's going to bind, and these are ion channels, right? They're channels for sodium that are ligand gated, right? A lot of the other channels that we talk about are all gated by voltage, right? As the action potential flows down, it's going to cause the opening of these channels. This one is a ligand gated channel because if you have acetylcholine bind, it will open a channel. Sodium's going to come in, and you are going to generate a potential, right? Basically, an action potential in that neuron, different, of course, from the action potential or action potential in the muscle, different
Hmm.
from a neuron, but kind of a parallel process.
Right.
Um, if you are triggering contraction, that action potential is going to travel deeper into the muscle through what is called a T tubule. And you're essentially going to trigger the release again of calcium.
We love that molecule.
I know. Acetylcholine and calcium really are the critical things to keep in mind when discussing neuromuscular junction, motor neurons, muscles, etc. Right? Like those are like the major players that are involved on multiple levels. So if you have that action potential travel down those T tubules, what it's going to do is pass right by what we call the sarcoplasmic reticulum. And that's going to sound very similar to an endoplasmic reticulum because it is. It's just specialized endoplasmic reticulum. And it's really specialized to store a lot of calcium. Okay, normally we think of smooth muscle as being, or sorry, smooth endoplasmic reticulum as being able to store a high amount of calcium. This is just specialized to store even more. Okay,
Mhm,
so
mhm.
if you do have that action potential travel down those T tubules, you'll activate ryanodine receptors and you'll have calcium flood out of that sarcoplasmic reticulum.
Mhm.
And ultimately, that calcium is going to trigger the next phase.
The next
Okay, so
chapter of our story, right?
yes,
Yeah,
and I
so
love that next chapter.
I will let you tell because again, I remember it's so funny. This was months ago when Molly was telling me the story, and I was like, "No way, this is the story that you used for." It's fantastic. I really am excited for you guys to hear this one. But yeah, 100%. Kind of just some I think super important things to keep in mind. These T tubules, right? When you kind of look at a picture of a muscle, right? So you have muscles. I always kind of like thinking of T tubules as these little holes in like the
Yeah,
membrane of the muscle that are
or
just extensions of the membrane because we know action potentials are just potentials on the membrane. Like they don't actually go deep into the cell of the
Mhm.
neuron. And because that's always a big thing about like why are two tubules important? Why can't we just do an action potential on the sarcolemma, which is just the plasma membrane of the muscle? Why can't we just do it there? But the problem is action potentials are only on the membrane. But we know if we looked at a picture of muscles, muscles are thick, especially if you're anyone like Molly, right? Really big muscles, right? Right? And we need that potential to penetrate deep into the muscle to activate all of those sarcomeres, which we'll talk about what those are, those contractile units. And so that's the purpose of those T tubules, which are just extensions of the membrane that just dive deep into the cell. And so that they'll kind of allow like little highways for the exponential to travel down deep in order to get to those sarcoplasmic reticulum, activate those voltage gated calcium channels and release all the calcium trapped in those reticulums into the sarcoplasm. I love how muscles are so like we just have to make everything sarcolemma,
Yes.
sarcoplasmic reticulum, sarcoplasm, like all of these things like that are just in a regular cell. If they have it, they just have to make it sarco-X, which I think is really, really, really fun. But yeah, now we've got all this. We've got just flooded a bunch of calcium into the sarcoplasm of this muscle cell, which enters the next chapter, and I'll let Molly take it away.
Okay, so chapter two began in the muscle. We have the action potential. The end of chapter two is calcium's released. Next chapter, chapter three. This one is my favorite. And I feel like we need a little context for this. So we're about to talk about how we actually have the shortening of muscle. Okay, so keep in mind if you are moving your body in any weird way, and if you can't see, I'm doing a weird little wiggle, right? If you're doing something
Mhm.
and you're moving your skeleton, you might see shortening and what you perceive to be lengthening. Like you could move your arm in different directions. But I want to be super clear. Muscles can only shorten, right? Like that's their job. They contract, they get smaller. You can move your skeleton around a lot because of different muscle groups that oppose each other. One may relax while the other contracts to create a lot of different types of motion. Okay. But at the end of the day, muscles contract. They don't create force through expansion. They contract and get shorter and then they relax back to normal length. That's what they're doing. And when we talk about how that works, it's all about actin and myosin. Actin's a microfilament. It's a cytoskeletal element. It is what it is, right? It's like a rope. Myosin is the motor protein associated with actin, and it can pull on actin like a person tugging on a rope, right? And that's ultimately what can create the shortening. We'll get into more of the specifics with that in a moment. But when it comes to chapter 3, which is how we even basically how can we grab onto the rope? We're not going to talk about the pulling yet, but if we are stimulated to contract, how can the hand and the rope interact at all? Okay, and this comes down to the idea of having voluntary movement. You don't want your body to be contracting without your input and direction, right? That can create tremors, that can create a lack of motor coordination, right? Like by evolutionary design, we want to have control of our movements, right? And so, unless we have a stimulus to contract, a neural stimulus, go ahead and contract muscle cell or motor unit, we don't want actin and myosin to be able to interact. Okay, so we actually have specialized, call them regulatory molecules that prevent contraction unless you are stimulated to contract. Okay, so if we think about the structure, you've got actin, picture it as a rope. Okay, then you have a motor protein, myosin, which wants to walk or grab onto the rope and pull on it. In order to prevent that from happening, we have a molecule. It kind of looks like a rope. You could visualize it like that, but it's
Like wrapping around it, yeah.
wrapping around actin and basically blocking all of the places where myosin, the hands, so to speak, could reach out and grab on. Okay. So you cannot have those two interact. Okay. That's the normal stage. And this is where my story comes. Mike, I told you this story literally
Long
like in
time
our
ago.
first couple podcast episodes, and he has been teasing me about it ever since. But you know what? It helps. So actin and myosin, these are our star-crossed lovers. This is a Romeo and Juliet story at the end of the day. Actin and myosin, right? They are held apart by fate, right? Unless there's a stimulus to contract, which we'll talk about. But actin and myosin cannot be together, right? And it's because in this situation, myosin has a superfan, right? Or like a stalker almost, right? This molecule is called tropomyosin. And you could remember that because it's literally so obsessed with myosin that it has stolen part of myosin's name, right? Tropomyosin is the molecule that will absolutely cover all of myosin's binding sites on actin. Basically, you can't interact. You have to interact with me. Look, I'm over here. I'm on actin. Don't you want to interact with me? Myosin's like, no. If tropomyosin is present and blocking actin binding sites or myosin binding sites on actin, there can be no interactions between myosin and actin. Game over. That being said, we do need the ability to contract sometimes. Okay. And we know the end product of our last chapter was calcium. So how can calcium move tropomyosin out of the way? Well, it comes down to another molecule, which I like to imagine as the youngest of 5 kids. This is tropomyosin's older sibling, right? So it's called troponin, and you can figure that out because it shares the family name of tropomyosin, right? Troponin, tropomyosin, they both share that tropo, right? Troponin, though, opposes tropomyosin. I like to visualize it as the older brother or the older sibling that, speaking from experience with 4 older siblings, they will not get involved with their younger siblings' drama unless they have to, right? If you bribe that older sibling with cash money, aka calcium, troponin will drag tropomyosin out of the way, right? It won't normally, right? Without the stimulus to contract, which leads to calcium release, troponin does not care. Tropomyosin, do your thing. I don't get paid enough for this, right? That's not my job. If calcium is present, troponin will yank tropomyosin out of the way. With those myosin binding sites on actin now exposed, it is possible for myosin and actin to come together, and we have finished our next chapter of the story.
Right.
So it's all Romeo and Juliet,
It's all just
basically.
Romeo and Juliet being held apart. Now they're together again because of tropomyosin the stalker. We love that. But yeah, so now, right, we've had the calcium being released from that sarcoplasmic reticulum. Calcium's there in the sarcoplasm, right? It is now on troponin, which is now moved tropomyosin. And now we can begin the next chapter of our story, which is the cross-bridge cycle, right? Which is the word that I don't know why I just spaced out at the beginning. I was like, cycle, which cycle is this? The cross-bridge cycle,
I
of
was
course.
like, how can I give you what you
Like
need?
I
I'm
was
like,
like, I was like I just completely blanked. But yes, the cross-bridge cycle, where now myosin has been waiting. Myosin's been ready. There was a stalker in the way, but myosin's been waiting and ready to grab onto actin. And this is kind of the first step of the cross-bridge cycle, is that myosin was in a high energy state. It was excited. It was ready to go. And as soon as that tropomyosin was moved out of the way, it can now bind and form the cross bridge. As cross bridge formation is the second step of our cycle. So now we have this high energy myosin. It's cocked. It's ready to go, binding to actin. It's binding to the rope. Kind of a note about this myosin, it's got the ADP and phosphate on it at this point. It's got those things on it. Those things aren't really doing anything now, but it's right kind of there, kind of in its back pocket. Now it's ready to go. And so now at this point, right, now it's grabbed the rope and it's high energy. This is when we have the power stroke. So imagine, remember, our actin's the rope and our myosin are kind of the hands. Now our hands are able to get rid of what was in the way, grab onto it, and actually pull on the rope. Right, in this process of pulling, right, it's going to be pulling it towards, and that's the actual contraction. Now we have to remember this is happening on a lot of different myosins, not just one little myosin head. A lot of calcium was released, so a lot of myosins are doing this together. This is why muscles contract. They've pulled it. But now, right, we just have this kind of pulled state. It's kind of grabbed onto the actin really, really tightly. And I was just like, okay, I'm pulled it. Now I can't let go. Right? I am just stuck on this actin. But this is where a really important molecule comes in, which is ATP. Now, ATP will come and actually bind to the myosin. And ATP binding to myosin is actually what tells it, yo, dude, let go. Right? Like, actually let go of that actin so that, hey, I can let go of the actin and actually get ready for the next pull. Right? Because by letting go of that actin, the myosin's now let go, and now we can use that ATP to get back into a high energy state again. Right? It's right to like, oh, I'm going to cleave that ATP, which again we know ATP, the energy unit of the muscle, right? That energy source. It can now cut it, you grab that energy and get in a high energy state again. It's now ready to go. Actin's there, and as long as the actin sites are still available, it's going to pull again, right? It's going to, as long as the tropomyosin's still out of the way, it's going to keep on pulling, and go through this cycle. The idea of like, it's cocked, we'll bind, power stroke, ATP, let go, cock, bind, power stroke, ATP, let go. This is the cross bridge cycle. It's why we call it a cycle, because it goes over and over and over again, unless there is something that could possibly stop it, which we can talk about later. But I also kind of want to leave a note where this is understanding the cycle is really important, because the MCAT loves talking about things like rigor mortis, right? Like, what is that? If you've ever had the unfortunate scene to see a dead body, as soon as somebody dies, you'll see that they tense up, right? All the muscles go really, really tense. Now you might be thinking, why? Well, if if a person is deceased, they're not producing any ATP anymore. So there is no ATP in the cell to tell the myosin, hey, let go. So it's just forever going to grab onto that actin, keeping it in a contracted state. So this is our kind of next chapter of like, when you are contracting, it's the cross-bridge cycle that's kind of going out, kind of over and over, keeping that contractile strength in our muscle cells.
I think the thing that is the most confusing about this is the ATP.
Yeah.
Like you're so used to, oh, the ATP binds
Hmm.
and then something special happens.
Hmm.
Like I think one of the most misunderstood aspects of this cross-bridge cycling, which you explained so beautifully, is ATP binding does not provide the power stroke. In fact, ATP binding allows you to let go.
Hmm.
We're so not used to that.
Hmm.
ATP, that's the high energy state. What's going on? Why would that let you release? Okay, it's unusual from what we think, but ATP binds, you let go,
Hmm.
you have hydrolysis of that ATP, and you get cocked again, and then you let that ATP or that phosphate and that ADP go, and you're able to grab on again, right?
Hmm.
So it's very backwards from what students intuitively think will happen, and yet I think it makes a fair amount of sense. But to anyone listening, I would keep track of that because
Hmm.
sometimes, when you get to a testing situation and you can't remember the exact details of it, you go with what's expected, what you would
Right.
think would happen. And that's one that I've seen, you know, when it's counterintuitive to most, those are the ones that you want to make sure you dive in deeper with and that you understand so that if you forget about it on test day, you don't default to what you'd expect,
Hmm. Right, right.
basically.
And that's why I love the rigor mortis example because like we've all kind of
Exactly.
heard of it. Like if you forget, like, oh wait a minute, what actually caused it to let go? I kind of just think back, well, okay, so rigor mortis is one. What's that person lacking? Oh, ATP. That's what actually
Right.
allows it to let go. That's always my kind of go-to to help me remember. But you're totally right because it doesn't totally make sense at first, right? Because you are thinking high energy. Um,
Yes.
and I think maybe it's important to step back here to actually talk about the kind of big picture of that contraction because I kind of mentioned before how we've kind of gone like pretty granular in terms of that
Yeah.
myosin binding to the actin pulling, but we have to remember it's not just one myosin. We've got a whole bunch of myosins and a whole number of actin binding sites that are kind of pulling simultaneously, and this is where I think it would be important to talk about the actual sarcomere structure. Now, when I say the word sarcomere, I always kind of like picturing the sarcomere as like the functional unit of the muscle. You know how we talk about functional units, like, neurons, the functional unit of the nervous system, the nephrons, the functional unit of the kidney. The sarcomere is the functional unit of the muscle. That's actually what gives it its ability to contract. Now, it's hard, it's going to be hard without a picture. And so maybe in post we can have like an editing like of like a hey, you wanted to post a picture of the sarcomere? Maybe we can get that in post. But when we talk about kind of sarcomere structure, there's important regions to keep in mind when we talk about the regions of the sarcomere. And so a great place that I like to talk about to start with are the Z lines, right? These are kind of like the lines when we talk about the striations in skeletal muscle. These are kind of those striation lines, right? Which actually are the borders of the sarcomere because the sarcomere has a length. On either border, there's a Z line, right? And then we have to remember there are two parts of the sarcomere. There's the actin part and there's the myosin part. Now, the Z lines are where the actin part originate from. So they're kind of like my hands, if anyone's watching, or maybe if you're watching, there's going to be a picture from the Z lines are where the actin extends from, right? So they have these kind of fingers extending into the middle towards each other, right? They're just kind of extending there. But again, actin is just the rope, like we talked about. It's the rope that myosin will pull on. And so any region that's just actin, actually, maybe I won't go to the bands. I'll talk about overarching, then I'll go in the bands, right? We then have the myosin part. Now, if the Z lines are kind of the ends and kind of extend to the middle, the myosin is actually starts in the middle of the sarcomere and extends out towards the Z lines. So you'll have these regions where actin's extending in, myosin's extending out, and you'll have these regions of overlap between them. It's in these regions of overlap that's where the pulling is happening. When they're next to each other, that's when the myosin's binding to the actin, power stroking, and pulling the sarcomere towards the center. The Z lines are getting towards the middle of the sarcomere. The sarcomere itself actually shortens. Now, this is the whole sliding filament theory. And when we actually look at like a picture of the sarcomere, now that we know the different kind of overarching ideas of actin extending the middle, myosin extending out, we can talk about the different regions of the sarcomere that we can actually see via, well, what's the imaging technique? Is it microscopy? Yeah, electron microscopy that actually usually we see that's the picture. Now, the first band is the A band. Now, I always think of the A band as the all band, right? The all band is like, hey, anytime you have overlap between the myosin and the actin, that means it'll be the A band. So it's essentially encompasses all the myosin and most of the actin, right? All the myosin and most of the actin because it's right in the middle. Because anytime there's overlap, you'll have the all band between myosin and actin. Then we have the I band, right? The I band I always kind of think of because I is like really thin. That's just so is actin. So that's the I band is only actin. Now I bands are always at the ends, right next to the Z lines, because again, that's we just only want actin there, so they're always at the ends, right? And then we have the H zone. Now I kind of think of the H zone as only myosin, right? We've talked about only actin being the I band. Well, the H zone is that part in the middle that's only myosin. Because again, the actin doesn't connect all the way into the middle. There's going to be a little region in the middle where it's only going to be myosin. That's going to be the H band. And when you contract, the whole idea of sliding filament theory that you've heard of is the idea of we've looked at how do these bands change, right? How do these bands change? And based on that change, what do we think is happening? Now, we've talked about the I band, H zone, A band. What we see, imagine when you if you start contracting, and anyone watching, you'll see me doing with this weird hand sign, where if you start contracting, right, your sarcomeres are going to get closer and closer together. So now, if they're getting closer and closer together, what do we think is going to happen to the bands, right? Molly, let me ask, if these are getting closer together, what do you think is going to happen to the I band, the region that's only actin?
So basically, we've got these different ropes extending to, let's say, the middle of a room, right? And then you've got people lined up on the middle,
Right,
right?
right.
That are all in the center of the room that are pulling on these ropes. The walls are closing in, essentially, right? The I band is the space between the space that's only the rope, right? If the
Hmm.
walls are closing in because people are pulling on those ropes,
Hmm.
the I band is going to get smaller
Hmm.
because there's going to be less and less rope that people are
Hmm.
not touching yet. Right.
Right,
So yeah, that
right.
will shrink. That will get smaller because we're pulling on them and it's getting closer. Actin and
Hmm.
myosin are overlapping more and more.
Right. Absolutely, because there's more and more overlap. There's now more people grabbing onto that rope. But I think that idea also holds for the H band, because remember, the H band was also the people that were kind of in the middle of the room that didn't weren't holding on to any rope yet. Kind of right, there's just myosin. Well, as you keep pulling the rope, that means the people at the end, the rope's going to extend to those people. So therefore, the region that's only myosin also shrinks, which is the H band that's in the middle of your sarcomere, in the middle of the room. So the regions that are only actin and only myosin will shrink, but the region that's the A band, the part of the overlap, stays the exact same, right? Because it's not like the people, think of the people have like, with the example, their feet are glued to the floor. The amount of area in the room, they're not like moving towards the wall. They're staying glued there. They're just pulling. So the region that's just people is staying the exact same, right? So we know the A band is the same. I band, H band, they will shrink upon contraction. And so it's this way that this is what we see as a result of our whole story, right? Of this contraction story is the shrinking of our sarcomere and actually what leads to all of these units contracting together.
Yeah.
Mhm.
So we've gone through almost the entire story now. Again, we
Mhm.
started with our motor neuron, chapter 1. Chapter 2 is the neuromuscular junction and
Mhm.
that communication between the two, right? Chapter 3, we had our muscle actual contraction. Then we went into the details of how to even facilitate or allow or permit actin and myosin to come together. That's the Romeo and Juliet. And then we had actual cross bridging, right? We're understanding the context. Really, the only thing left to talk about here is how does this stop?
Right. Imagine if I just contracted and I'm just like, oh well, I'm stuck like this
Yeah.
now.
That would be super, super dangerous. That's like what we talk about with tetanus, right?
Right.
You don't want sustained contraction, just constant force. You want to be able to let go, right? This
Mhm.
is exhausting, right?
Mhm.
It actually can create a whole bunch of other host issues later on. Please, right? We want to be able to relax. So,
Right.
how does this work? How do we let back up? Basically, we're just undoing what we did to start it. Okay.
Mhm.
We know that the ultimate signal that came into the muscle cell to allow myosin and actin to come together, the bribe to have troponin pull tropomyosin off of the myosin binding sites of actin, facilitate cross bridging. What was that bribe again?
Calcium. That was the bride. That was calcium, right?
That was the bribe. It's the cash money, right? It's
Cash.
calcium. So if calcium being present is what will allow troponin to pull tropomyosin off,
Mm,
a great way to stop contraction is actually just to take the calcium back, right? If the calcium is gone, troponin being the older sibling, it's like if I don't physically have to be here, if I'm not getting paid to be here, I'm not messing with my little siblings' drama, right? I'm backing out. You do whatever you want. I don't care. I'm not paid enough for this, right? You take calcium out and troponin stops. Tropomyosin will now go back into its sort of blocking formation where no cross bridging can occur. And that's a huge stimulus. So if we want to stop contraction, just take calcium back. Okay. And if we are talking about taking calcium and putting it back where it came from, we're going to be pumping it back into the sarcoplasmic reticulum. Now, one thing that we might wonder is this an active process? Is this a passive process? And how can we tell?
mhm,
Right. But we have to think action potentials as they flow through that muscle, as you have the wave of depolarization coming down the T tubules, creating the release of calcium, that should be passive, right? It should be passive. We should be able to just open some calcium channels in that sarcoplasmic reticulum and have a huge flood of calcium. You don't want to have to pump calcium out because it's slower. We want our muscles to be fast in responding to our stimuli, okay, to contract. So if it's passive for calcium to suddenly flood out of the sarcoplasmic reticulum, what that implies is there's probably a lot more calcium in the sarcoplasmic reticulum than there is in the cell interior. And again, we know that because calcium is, if present, is going to allow us to cross bridge cycle. So if we want to put calcium back where it came from, that has to be an active process. We now have a lot of calcium in our sarcoplasm, which is cytoplasm with a fancy name. We have to pump it back into the sarcoplasmic reticulum, which is endoplasmic reticulum with a fancy name.
mhm.
We want that to be an active process so we can create a highly concentrated sarcoplasmic reticulum with respect to calcium so that the next time that motor neuron or that muscle cell is triggered to contract, it's got a wave of calcium that it could release again.
Mhm. Yeah, no, this active process, right? Because I always kind of like think of the sarcoplasmic reticulum as like the storage unit of calcium. Like there is so much, they jam so much calcium into that place. Or even if we released some calcium, because I've seen students think like, oh, we've released the calcium. There's no calcium in the sarcoplasmic reticulum. No, in relation, when we release it from muscle contraction, we only put like a drop, like we only like put a little bit of calcium in respect to how much is actually there. So there's still, even after we activate, actually contract the muscle, there is still a ton of calcium in the sarcoplasmic reticulum. So even though we're kind of putting it back, no, it's still a very active process, and therefore active process means we need ATP again. So ATP serves a very, very important function in muscle. It's a very active process, contracting a muscle, right? You need ATP to actually bind to the myosin to release the actin to cock the head, but you also need it in order to actually get that calcium back into the sarcoplasmic reticulum. But now I'm thinking, wait a minute, there's no more cash money. There's no more cash money. So now so now wait a minute. No more cash money means troponin, the older brother, is like, I'm gone. I am not. And so by troponin not now pulling little brother tropomyosin out of the way, tropomyosin is now free to do what a stalker does, right? To kind of to kind of go back over those myosin binding sites on actin and be like, hey, myosin, look at me instead. Right? Now
Yeah.
it's stopping the myosin from binding to the actin. So again, it kind of like interrupts that cross bridge cycle. Because remember, right, the myosin came off, right, in order to cock its head, but now it's like, wait a minute, tropomyosin's back. I can't bind to actin anymore. So now I can't contract anymore. I've got this stalker here, right? I can't actually bind. And so because the myosin cannot bind anymore, there's no more contraction, and that's where the muscle actually relaxes, which is kind of like the end of our kind of the end of the book, right? We talked about how it actually leads to contraction and relaxation. Now, if we want to start again, we'll start back from the top with the neuron. With if I need to contract the muscle, we'll do the whole story once again.
I love telling these complex processes like stories. It's so much fun because if you study each area independently, it's so much harder to understand the broader picture. So when Mike and I were talking about what we wanted to cover in our next episode, we were like, okay, this storytelling aspect is it's so fun on our end and hopefully so helpful for you guys. And we're like, what other kinds of processes are really confusing unless they're told in a story? And in our next episode, or really next two episodes, we're going to tell maybe one of the most complex and hated, not
True,
hated, but
true.
but things that people don't like learning about, which is essentially the combo of gametogenesis and how we time meiosis, how that's different from mitosis. Like, how do we create gametes? How does the menstrual cycle work? How do we time it throughout our life, which
Mhm.
can be scary alone? And then from gametogenesis, how do we have fertilization and how does that early embryo develop into a future human being? So we've got like one of the ultimate stories to now be told.
Truly, the story of where we all kind of came from,
I
really.
know.
And I'm so
Uh,
happy that we're going to talk about that next episode because I know that it's actually an area that really gets overlooked, I feel a lot. Like
yes.
people really skim that area, but it's really one, there's a lot of really testable material there that's like
Mhm.
really good content they can test on. So that's why I'm really happy to be explaining it also just not in a way to memorize. Not just don't just memorize the process, actually understand the why, understand the story of how a zygote and eventually baby comes to be. So I'm very excited to be talking about those things next episode. So wherever we're finding you, wherever you are in the world, whether it's day, night, you're on the train, missing a train possibly, right? We hope you all have a fantastic rest of your day and happy studying, everybody.
Happy studying.
Bye.
Bye, everyone.